Hello and welcome to Bio40 Pre Lecture 19. Today we're going to cover transcription, how mRNA is copied from DNA. Once again, we return to the central dogma of molecular biology, the flow of information from the genetic molecule DNA to the messenger molecule RNA to the functional gene product, proteins. The process of a protein being created based on the information in the DNA is called gene expression, and there are two main steps. The synthesis of mRNA from the DNA template is called transcription, and the mRNA is read and used to make a polypeptide chain in the process of translation. We're going to go over both processes in the next few lectures and address how each differs in prokaryotes and eukaryotes. We'll begin with transcription. Transcription is analogous to replication in many ways, though you do need to be aware of several important differences. Let's start with the polymerase. Just as DNA polymerase was the key enzyme for replication, RNA polymerase is the key enzyme for transcription. Like DNA polymerase, RNA polymerase is actually a large complex of proteins. The E. coli version has 5 subunits. 4 of them form the core of the complex, but there's a 5th interchangeable subunit called the sigma factor. The sigma factor plays an important role in initiating transcription, but is unnecessary after that. In yeast, RNA polymerases are even larger. Yeast RNA polymerase II has 12 subunits. Its crystal structure is shown here on the left, with each subunit in a different color. RNA polymerase extends a new RNA chain in a manner that should remind you of DNA polymerase. A DNA strand still serves as the template, though instead of dNTPs, NTP building blocks are used to build the new strand. So if A is the next base in the template, RNA polymerase will bring in a UTP to pair with that A, and it will catalyze a reaction that links the first phosphate of the UTP to the 3' hydroxyl of the previous nucleotide in the chain. This is the formation of a phosphodiester bond. Just as cleaving the triphosphate bonds of the dNTP building blocks provides the energy for replication, the triphosphate bonds of the NTP building blocks provide the energy for transcription. Unlike DNA polymerase, RNA polymerase doesn't need an existing 3' hydroxyl to begin the chain, so there's no need for a separate enzyme to start the process with a primer. But RNA polymerase will still always proceed in the 5' to 3' direction. Now let's walk through the 3 main parts of transcription: initiation, elongation, and termination. First, initiation. Upstream of the point where transcription begins, there's a DNA sequence called a promoter. Upstream is used to refer to something being in the 5' direction relative to a certain site, while downstream means in the 3' direction. In E. coli, the promoter has 2 parts, a -10 and -35 region, so called because they are about 10 and 35 bases before the spot where transcription begins, which is called the +1 base. Transcription is initiated when the sigma subunit of RNA polymerase binds to the promoter. 10 different bacterial promoters are shown here. Note that the -10 and -35 regions are similar but not identical. If you take the most common base at each position, you get what is called the consensus sequence. In general, the closer a given promoter sequence is to the consensus sequence, the better it will bind with the sigma factor. Better binding leads to increased transcription of the genes that follow, so these are called strong promoters. So as the first step, we have the RNA polymerase binding to the promoter. This full complex, the core with the sigma factor, is called the holoenzyme. Now specifically, the sigma factor binds loosely to the -35 region of the promoter. Next, the DNA is unwound and separates starting at the -10 region. A helicase is not required for this. The spot where the DNA strands separate, which resembles a replication fork, is called an open promoter complex. Transcription begins when the polymerase starts adding RNA nucleotides from the +1 position. Now we are on to elongation. RNA polymerase extends the RNA chain in the 5' to 3' direction, just as we talked about earlier. After about 10 nucleotides, the sigma factor falls off. Its role in initiation is done, and only the core enzyme is needed past here. The new chain is extended at a rate of about 30 to 50 nucleotides per second. You can see some key differences between replication and transcription here. Transcription is unidirectional and not bidirectional, and only one strand is copied. There is no lagging strand and no Okazaki fragments that need to be ligated together. As transcription proceeds, you temporarily see a DNA-RNA hybrid molecule with the second DNA strand left unpaired. But later, the RNA will separate from the DNA. It will function as a single-stranded molecule, and the two DNA strands can rejoin. The process continues until the polymerase reaches a terminator sequence. There are two types of these sequences, row-dependent and row-independent. In row-dependent termination, the row protein halts the process. This protein binds a specific RNA sequence, so that sequence needs to be transcribed before termination can occur. After row binds its site, it catches up to the RNA polymerase and forces it off the DNA. This requires energy in the form of ATP. However, termination does not always require an extra protein. Row-independent termination uses a sequence called an inverted repeat, shown here in pink. Reading the top strand of the first pink sequence, 5' to 3', is the same as reading the bottom strand of the second pink sequence, 5' to 3'. The resulting RNA transcript can base pair with itself. When it does, you get this stem loop structure that is called a termination loop. The termination loop is followed by a repeating series of uracils. The repeating uracils slow down the RNA polymerase, allowing the termination loop to interact with the protein complex, which causes it to fall off the RNA. So again, the termination sequence is transcribed before it actually causes termination. Researchers have shown that mutating the sequence so that a stem loop can't form prevents termination from occurring. You get the same effect by deleting the poly-U region so that the polymerase doesn't slow down. Those are the basics for prokaryotic transcription. Now let's look at some ways eukaryotic transcription differs. Eukaryotes have several RNA polymerases, and each specializes in a different type of RNA. mRNA transcription is carried out by RNA polymerase II. Eukaryotic polymerases don't rely on sigma factors to bind promoters. Rather, there are other proteins called transcription factors that bind the promoter or other nearby regions of DNA and recruit the RNA polymerase. Finally, in prokaryotes, the RNA molecule as it's transcribed is the final mRNA. But eukaryotic transcripts undergo processing before they can be called mRNA. There are three main types of processing the primary transcript undergoes. The 5' end is capped with a methylated guanine. A polyadenosine tail is attached to the 3' end, and regions of the genes called introns, which are not part of the protein coding sequence, are removed or spliced out. This image shows the methylated guanine attached to the 5' end of the transcript. You can see it's attached backwards. The two sugars are linked from 5' carbon to 5' carbon via a triphosphate bridge. Also, it's not just the guanine that's methylated. A methyltransferase adds methyl groups to the 2' hydroxyls of the first two sugars of the transcribed nucleotides. This all stabilizes the mRNA molecule, preventing it from being degraded too quickly. And it plays a role in ribosome binding, which is key in the next step of expression, translation. Next, we have the adding of the 3' poly-A tail. The last part of the transcript is removed by a ribonuclease, and an enzyme called a poly-A polymerase adds 50 to 250 adenine nucleotides to the end of the chain. This, too, is thought to stabilize the mRNA molecule, as well as assist in transporting it from the nucleus to the cytoplasm. Finally, let's talk about splicing. While prokaryotes are highly efficient with their DNA, eukaryotic organisms are considerably less so. If you look at our entire genome, that's the whole collection of our DNA, you'll see less than 2% actually codes for proteins. We can talk about the other 98% another time, but the key thing to know for now is that some of these non-coding regions are interspersed between the coding regions in our genes. We call the coding regions of genes exons and the non-coding regions introns. And some genes have a whole lot of introns. One of the most extreme examples of this is a human dystrophin gene. The primary transcript for this gene is about 2.5 million nucleotides. That's larger than the entire genomes of some bacteria. The role of splicing is to remove the introns, leaving just the protein-coding exons. And after you remove the introns from this gene, the final mRNA is only 14,000 nucleotides. The vast majority of this primary transcript is non-coding. Not all cases are this extreme. A small minority of human genes don't have any introns at all. But most transcripts require at least some splicing out of introns. Splicing is regulated by short sequences around the exon-intron junctions. At the 5' end of the intron, you have the splice donor site. A little further in, there's a branch site. Then at the 3' end, there's a splice acceptor site. Splicing occurs in several steps. First, there's a cut at the donor site on the 5' end. The free end curves in and binds the branch site, forming a loop that is sometimes called a lariat. Next, the acceptor site at the 3' end of the intron is cleaved. The adjacent exons are brought together, and the intron is degraded. While this might sound pretty direct and simple, it's really fairly complex. Large machines called spliceosomes help mediate the process. They consist of around 50 proteins and several short RNA molecules called small nuclear RNAs, or snRNAs. So it ends up, this isn't really the whole story for splicing. There's also a phenomenon called alternative splicing, where different mRNAs can be made from the same primary transcript. I alluded to this a few lectures ago when I said multiple polypeptides could be encoded by a single gene. Take this gene that codes for part of an antibody. Some antibodies are found on the surface of cells, and some are secreted into the blood. The difference comes down to alternative splicing. This gene has 8 different exons. There is a splice site in the middle of exon 6. If that site is spliced to exon 7, you get a membrane-bound antibody. But if the site is skipped, the transcript ends with exon 6. Exons 7 and 8 aren't used, and the antibody is secreted. There are several ways alternative splicing can work. An exon can be included or skipped entirely. You can have alternative splice sites where the donor is always at the same position, but there are 2 different splice acceptors. Or you could have 2 different splice donors that both use the same splice acceptor. And finally, there's intron retention, where the intron just isn't removed at all sometimes. It's interesting to note that different organisms use these different forms of alternative splicing to greatly varying degrees. This table compares usage in humans to the common model plant Arabidopsis. That does it for transcription. To sum up for eukaryotes, RNA polymerase II forms an RNA copy of the DNA sequence called a primary transcript. The transcript is modified with a 5' methylated cap and a poly A tail. Then the introns are removed by splicing, and alternative splicing means sometimes multiple final mRNAs are possible. The mRNA is now ready to be translated to a polypeptide chain. But that's for next time.